All right. In the interest of time, we'll go ahead and get started. So this is the Symposia 7, Game of Thrones, Inflammation, Initiators, and Drivers Across the Lifespan. We thank you all for being here. I'm having the pleasure of co-moderating this with Christine Bojanowski, who's an associate professor at Tulane University School of Medicine. And my name is Dan Cook. I'm an assistant professor at the University of Iowa. And we're really excited to talk about this topic, which is really something that is somewhat underappreciated in our epithelial-centric field, is the contributions of both the innate and the adaptive immune systems to CF disease. And we're hoping to explore that more. Here's our learning objectives for this session. But we have a whole host of speakers here that are going to really talk about multiple aspects of both the innate and adaptive immune system. Our first speaker is going to be Nick Klimuk, who is going to talk about initiation of inflammation infection using some data he had from preschoolers and pigs. Unfortunately, he is not here today, so this will be a recorded talk presented. This will be followed by Camilla Margaroli, and then we'll have Emanuela Rashia and Clemento Rito speak following that. So with that, we're going to go ahead and get started. I will introduce Nick's talk here. See if we can get it going. Okay. So Nick Klimuk is an associate professor at the Large Animal Models and Cardiovascular Research and Clinic for Cardiology at the Technical University in Munich. He really has focused on large animal models to understand monogenetic contributions to them with ideas of developing therapies. He recently had a publication in Science Translational Medicine where he was looking at data derived both from preschoolers at early time points before the onset of infection and comparing this with some of the findings he was having in pigs. So I will start his talk now, and thanks, Nick.
Dear Chairman, many thanks for the introduction. Dear ladies and gentlemen, my sincerest apologies for not being able to attend the conference in person. I will still do my best to introduce you to the topic. I have no conflict of interest, and I want to start with a few words on pig models in CF research because they are not so frequently used. This is quite surprising because the way of how a genetic defect in the CFTR channel develops into multisystemic and multifactorial diseases in patients is still unclear. And so we clearly see a relevance of pigs to be implemented in a bunch of complementary models to eventually improve the patient's quality of life. The generation of the pig model for CF itself is not a very new story. They have been developed 15 years ago or even earlier, but they show some striking features. First, there is microgallbladder. Second, at the disruptive male reproductive tract. Severe fibrosis in the pancreas, mild malformations in the airways, and a striking feature is the almost complete abundance of intestinal obstructions in the pig models. In the US and in Europe as well, the CF models have been used in the past decade for quite intensive research on various topics. But today, I want to focus on the immune system, or rather a relevant question, is there a predisposition for immunological alterations? Mostly this is summarizing a publication that was out since early 25 and investigating pigs and preschool children as well with complementary methodologies. We were facing quite complex challenges when we started the topic, because evidently local proliferation and maturation of immune cells in the periphery may be influenced by environmental stimuli, but also from alteration in hematopoiesis and different maturation processes in the circulation. Blood is the only specimen that is accessible in humans as well as in pigs. the other tissues were examined in pigs alone. The second layer of complexity was the understanding of immunology, because if you're talking with clinical researchers, they will discuss with you epidemiology, therapy, vaccination, personalized medicine, etc. In contrast, basic researchers love to tell about humanized mice, detailed mechanistic studies, genetically modified models, and so on and so forth. And pig immunologists have a very different focus, namely agricultural research. And there comes in economy, livestock, proliferation, vaccination, and so on and so forth. And for this reason, we carefully took out our study, first recapitulating the anatomy of the lungs, which appeared grossly normal in the entire cohort between CF and wild-type animals. Also very similar were the levels of cytokines in the airways of CF animals and wild-type animals, and proteomics, bulk proteomics on lung tissue was indicating almost no differences between wild-type and CF. When we dug deeper, however, examining mononuclear cells derived from the lung, we saw a quite striking feature, namely a strong evidence for granulocytic proteins in these mononuclear cells, indicating neutrophil identity. However, when we examined blood counts, we did not see any evidence for polymorphonuclear cells. What we saw in contrast, however, is that these mononuclear cells were enriched in the lungs of CF animals, but not in the spleen and not in the blood. The identity of these lung cells was unclear because adaptive immune cells appeared grossly normally. However, when we examined myeloid cells at histological levels, we saw an enrichment of myeloid cells in the interstitial space, mainly in the upper airways. When we explored it in more detail with conventional flow cytometry, we saw that these mononuclear cells almost completely showed myeloid identity, and we saw a slight differentiation of CFTR cells from wild-type cells in these mononuclear cells from the lung. This different pattern mainly arose from CD14 pattern and Ki67 pattern here in the TSNE plots, but also expressed at quantitative levels, revealing significant differences of CD14, but also of CD14 negative cells. When we aimed at the molecular changes at cellular resolution by a single-cell RNA sequence, we saw the appearance of quite distinct cellular patterns, and within major clusters, we again saw a different localization of CF and wild-type identities. This was particularly true in major clusters. For example, in fibroblasts, their appearance in mononuclear cells was to some surprise, but we've also faced some other unexpected findings, namely the appearance of a small fibroblast cluster that was not predicted by RNA-seq databases, but upon consultation on developmental biology papers, it appeared like cartilaginous fibroblasts. Even more strikingly, both fibroblast clusters differ between the genotypes in the collagen expression. So effectively, collagens were downregulated in CF, indicating some relevance in remodeling and fibrosis. We did not investigate this in more detail, but focused on the immune cells. Again, indicating differences between wild-type and CF, and revealing 4 major clusters, 1, 5, 3, and 12. 5 being clearly macrophages, 1 very likely pulmonary monocytes. 3 and 12 were some interesting clusters because they appeared almost exclusively or dramatically enriched in CF and were almost not abundant in wild type. On consistently regulated genes, they showed identity with cluster 1, but they showed no similarity between them, suggesting that they were enriched and unrelated myeloid populations in CF. And indeed, when we looked at classical marker genes for myeloid cells, cluster 13 turned out to be a strongly proliferating cluster, indicating tissue residence, whereas cluster 3 had clear signatures of monocyte-like cells. For this reason, then, we turned to PBMCs, both in pigs as well as in human beings, processed them similarly by single-cell RNA-seq, and identified, not to too much surprise, a more divergent picture of adaptive immune cells in preclinical preschool children compared to newborn pigs. However, the myeloid cells revealed a very similar pattern. First, they had the classical monocytes, intermediate monocytes and non-classical monocytes. The classical monocytes had a signature that was almost identical between human and pig and identical to this cluster 3 in the lung-derived mononuclear cells. And second, we also saw the splitting into CF cells and wild-type cells. When we did a pairwise comparison between those at transcriptional level, we saw a strong upregulation in CF cells by ribosomal-related proteins, whereas the same ribosomal proteins were upregulated at proteomic level, but additionally, we saw this recapitulation of granulocytic proteins as well. At the functional side, we explored the capacity for phagocytosis in CF and wild type. We saw that cells expressing typical scavenger receptors like CD11b were not much different, but the expression level of the scavengers was generally more declined in CF than in wild types. When we did phagocytosis assays with fresh blood cells from pigs, we saw that almost all of the myeloid cells were capable of ingesting bacterial particles, but the amount of particles was declined in wild type in cystic fibrosis cells. With ROS, the picture was even more significant. We had fewer cells capable of producing ROS, and the expression level of ROS was declined. When aiming at cross-species comparison of the phagocytosis potential, we faced some logistical challenges, namely the systematic blood sampling in human patients. And so we purified the cells, cryopreserved them, and only did systematic experiments after a cohort has accumulated. We did this in human and in pig likewise to facilitate comparison. And there we saw both for Staphylococcus aureus and Pseudomonas aeruginosa a declined phagocytic potential of human classical monocytes, also of pig classical monocytes, and also of myeloid cells derived from the lung in pigs. So we had quite a clear correlation of altered molecular signatures developing towards an altered functional profile, but two questions were remaining. First, what explains the distinct molecular profile in transcriptomic and protein levels, namely the enriched granulocytic proteins? And what is the correlation of these findings to CFTR expression? For the molecular profile, it is quite evident that myeloid and neutrophil lineages are relatively strictly separated, and that granulocytic proteins are produced in a highly orchestrated and timely staggered manner. Only since recently, there is some evidence of emergency cases, and in these cases, the classical conventional monocytic line is fading, and instead, monocytes are derived from the neutrophil lineage by transdifferentiation. And this transdifferentiation starts after granulocytic proteins are being produced and their transcription is switched off. So explaining these granulocytic pattern at protein, but not at transcriptional levels. We recapitulated this also in the bone marrow. When performing single-cell RNA-seq in bone marrow of newborn pigs, we for sure faced a quite immature picture of bone marrow populations. Still, we were able to correlate major clusters to established populations, and we identified cluster 1 as bone marrow-derived monocytes upon their classical pattern, but identified their progenitors as the source of these granulocytic proteins like ASU1, PRTN3, HP, and so on and so forth. So giving evidence that in CF we see emergency myelopoiesis creating these granulocytic myeloid cells. The last question is a bit controversial, I have to admit. The question for CFTR expression in immune cells is compromised because the literature is ambiguous on CFTR expression. One of the major reasons is that CFTR is generally lowly expressed in native tissue. Effectively, we and others were only able to detect CFTR protein in classical histological sections in the intestine, but not in the lung. More sensitive immunofluorescent assays and single-cell RNA-seq assays clearly reveal why this is the case. First, you have a very, very small population of CFTR expressing monocytes, and second, the expression of CFTR in there is relatively low. And the second challenge we faced is that obviously transcriptional regulation in immune cells is highly dynamic. So it is quite unclear at which stage CFTR would be expressed and whether cultivation may affect aggregated CFTR expression. So for this reason, we focused on freshly isolated native tissue, and there we saw expected CFTR expression in the major CFTR organs, lung, intestine, also a bit in the liver and so on, but CFTR transcript levels were below threshold in all immune cell populations examined. So to come to a conclusion, we have a clear molecular phenotypic and functional change in the myeloid cell population in CF, which we correlate to an emergency myelopoiesis in the bone marrow. We see that the alterations in these fresh immune cells in the pig are not correlated to CFTR expression. So we formulated alternative hypotheses, two quite distinct ones. So first, CFTR may play a role in the shaping of the immune system during fetal development, but not later on. And so an imprinting effect, a push effect that constitutes in the bone marrow. Or we may face emergency myelopoiesis due to stress conditions in peripheral organs like the lung, which would constitute a pull effect. And with this, I will come to an end. Thank our extensive collaborative network that allowed us to compile this study. And thank you for your attention. Thank you very much.
right, I guess. Ooh. Gotta make the room explode. Can you all hear me? So I have a disclosure. I'm funded by Vertex through the CF Research Innovation Award. So thank you all for being here. Thank you, Dan and Christine, for inviting me. When I first got the email that I was invited to talk, I didn't know what I signed up for. I was asked to talk about host pathogen interaction in chronic inflammation in CF, which, as you know, is a huge topic. And I was given 20 to 25 minutes. So thankfully, I like a good challenge. So hopefully today I'm gonna inspire you guys to think about host pathogen interaction in maybe a new light that you haven't thought about. And for those of you that are fans of Game of Thrones, you may see some nuggets throughout the talk. So I started working in CF almost 10 years ago now, when I started my PhD in Dr. Tiruvan's lab at Emory. And the first thing that we did was writing a review article in which we described how neutrophils help shape the ecosystem that is the CF lung, and how they do this with relationship with the epithelium, with the pathogens that are present and that are colonizing the lung, but also the relationship with other immune cells and how that promotes chronic inflammation. And even though the face of CF during the last 9 years has changed dramatically, I can say that some of these ideas may still hold true today. We still see chronic inflammation and we still see infection in the CF lung in spite of ETI. So the way that I'm thinking about host pathogen interaction is that it's not a static crosstalk. It's actually evolving over time. It's starting from newborns and possibly in utero. And then it moves on as, our patients grow old. And there is an onset and there is an evolving phase. And now we're going to see how also aging is impacting this coexistence. And throughout the lifetime, there are basically episodes of exacerbation that also could change how the host interacts with the pathogens. So I don't have a whole lot as far as mucosal interaction in the gut, but hopefully there are a few references here and there for those of you that work at the mucosal level in the gut. So the way that I see the infection is there are 4 big categories where we have our own microbiome, the colonizing bacteria that are promoting chronic infection, the fungi, and viruses. And how they all interact with each other and the immune system complicates matters even more. So a few years ago, there was a really good review that thought about how these commensals and pathogen interacts with each other in the lung, and how there is a climax population and an attack population, and how this promotes both resilience and adaptation to the environment. However, I would like to challenge this in a way that we're still missing a few components, including viral infections and how the immune pressure is shaping this evolution over time. So what are the players from an host perspective? And I don't want to take thunder away from Manu for her next talk, but so we have the resident macrophages in the lung. We know that they are hyperinflammatory. We know that they are suboptimal phagocytes. And then a few years ago, we showed that neutrophils themselves change dramatically when they migrate from the blood into the lung. And this is dependent upon a transcriptional adaptation, which relates to their ability to actually clear bacteria. And recently, we added another piece of the puzzle where we see a switch in how the neutrophils deal with certain categories of pathogens, whereas they reduce their bacterial clearance, but they are increasing their ability to clear virus in the lung. And as far as adaptive immunity, we know that there is a predominance of Th17 responses. This promotes neutrophilic inflammation. We don't know exactly how that relates directly with pathogen colonization. However, we know that a B cell response is blunted by proteases that can cleave antibodies and therefore promote chronic infection. So one of the first strategies, and we heard also a really nice talk this morning about how Pseudomonas, for example, adapts to survive into the lung, how mutations in the O antigens can promote its survival and reduce phagocytosis by THP1 cells. Last year, there was a really nice publication that showed that changes in Pseudomonas LPS basically promotes immune response, and that is not modulated by ETI. But also there is work that shows that Pseudomonas can actually express sialic acid on its surface, and this combines to cyclic molecules on the immune cells, and that dampens the immune response. But also, in addition to how the pathogen adapts to the lung, is also what is the role of commensals in the microbiome. And although there is a lot of variability, which is a huge issue, I would say, or a good thing, basically individual variability, besides sampling variability, and also how different countries are applying treatment to the patients, can generate a lot of diverse results in what is truly important in the microbiome in our patients. A few years ago, Marcus Small's group basically showed that a reduction in Shannon index was correlating with basically increased inflammation in the lung. So another study that addressed directly the role of commensals showed that basically there are certain species of Streptococcus that can reduce IL-8 production by epithelial cells. And they show this in vitro, and they show this in vivo. So presence of certain bacterial species in our microbiome can actually prevent chronic inflammation. And not only that, they went on and showed that they don't only prevent chronic inflammation, but they can also dampen Pseudomonas aeruginosa infection. So they did this in co-culture, and then they went on and identified a molecule that is being secreted by our commensals, which is acetic acid. And that could actually reduce Pseudomonas growth. So our microbiome is important. I think there are more studies that are needed to truly understand the diversity and how that plays a role in the evolution of chronic infection in the lung and how it shapes the immune system itself. So one other factor that our microbiome releases is short-chain fatty acids. Mainly has been studied in the gut, but we know that these short-chain fatty acids can actually travel into the lung. A few years ago, there was a study that showed that acetate actually is really important in shaping the resident macrophages response in basically preventing RSV infection. And this will become important a little later on in the talk on how we shape the antiviral response. However, in CF, we don't really know still how short-chain fatty acids shape our inflammatory response. They can signal through G protein coupled receptors, and they can elicit either pro-inflammatory or anti-inflammatory signaling depending on the pathway that they activate. So that brings me to the lung-gut axis. I know it's a novel topic and it's still very understudied, or it's very also complex. But I think that it deserves a thought when we think about how the hosts respond to chronic infection in the lung. There was another talk this morning, which I'm really happy about, that we're starting to see some more immunology, how basically the microbiome in the gut shapes the activation of the immune system and how does that relate to what's going on in the lung. So I think that even though we're just scratching the surface, it's worth future investigation. So it's not only short-chain fatty acids that can shape the immune response, but we also have an imbalance of long-chain fatty acids in CF. It's known that omega-3s are reduced, and we see an increase in linoleic acid. And how does that affect our immune response is still under investigation, and is still not clear in CF exactly what are the true effects of that. However, it's been shown that in neutrophils, long-chain fatty acids can induce release of granule proteins, including proteases, but also they can make them undergo mitosis. So how do the pathogens exploit this dysregulation of the neutrophil adaptation? So in CF, we see a reduction in phagocytosis and a reduction in bacterial clearance. However, we see an increase in netosis and an increase in exocytosis of neutrophil elastase, especially rich granules. So how do the pathogen exploit netosis? There has been study, not in CF, but in other instances where they show that staph actually can cleave the DNA itself and use it as a fuel to promote growth. So whether that is actually happening in the lung is still unknown, but I think that we can draw parallels between other studies. On the other hand, exocytosis, especially of neutrophil elastase, has been shown to repress flagellin expression in Pseudomonas. A few years ago, we showed that uptake of neutrophil elastase by macrophages induces expression of PD-1, which is a surface protein that you may have heard of in cancer, but is also present in the CF lung, and that is directly related to the impairment of bacterial clearance by airway macrophages. And then recent work by Jonathan Ma showed that also neutrophil elastase itself can impair phagocytosis in the macrophage population as well. Now, elastase not only cleaves or changes Pseudomonas and cleaves other substrates, but it can also cleave IL-22. So IL-22 is a cytokine that promotes epithelial health and epithelial proliferation, is induced by short-chain fatty acids. So our commensals actually play a role in this. And in the CF lung, or in CF itself, we see that there is an increased expression of its receptor antagonist. So in addition to IL-22 cleavage by elastase, there is also blockade of the signaling pathway by the receptor antagonist. And not only that, in a separate study, it was shown that amount of IL-22 actually correlated with lung damage, which is contrasting a little bit. However, there was a study that was done in the skin itself, showing that IL-22 by itself can promote epithelial growth. However, IL-22 and IL-17 in combination can delay wound healing in presence of Pseudomonas and staph. So we know that the CF lung is enriched for IL-17 as well, in addition to IL-22. So I think that the role of these two cytokines in concert and how it relates to presence of bacteria and also wound healing should be further explored. So that brings me a little bit off topic, but it's going to tie in a second. With regards to wound healing, we see a shift of the immune system towards promotion of wound healing in the CF lung. We see in our own results that there is activation of the EGFR pathway. We find the ligands in the sputum. We found that neutrophils can actually express these ligands and release them. And we find that EGFR can actually be transferred to the epithelium via extracellular vesicles. So what if our immune system is actually tuned towards a pro-repair and antiviral mechanisms at the expense of an antibacterial and antifungal performance? So I think that there are some data from us and other groups that suggest that that shift is actually present in the CF lung. And that leads me to the work of Sebastian Riquelme and Alice Prince on itaconate. So itaconate itself is being shown to actually be released by macrophages and improve mucociliary clearance in the epithelium. However, in CF, it gets exploited by the bacteria to promote Pseudomonas adaptation and survival within the lung. Similarly, through a really elegant study, they did metabolite tracing, and they found that not only it allows Pseudomonas to adapt to the lung, but it also promotes biofilm formation by staph. So hopefully, they will continue on this work from thinking also about the wound healing perspective and mucociliary clearance, not just from a host pathogen interaction. And that leads me to the last topic. I don't know if I'm over time or not, which is also a very novel topic that I think it's worth future investigation, and that is the role of extracellular vesicles and outer membrane vesicles. It's been shown previously by Dr. Stanton that basically bacteria can actually release these OMVs that acts on the airway epithelial cells and basically reduce neutrophil recruitment. However, this crosstalk, it needs to be further investigated, as I would suspect that this population is very heterogeneous as well, and how these vesicles actually may promote tolerance from the immune system towards specific bacteria that are in the lung. We know that they are rich in proteins and they're rich in genetic materials. However, how they shape the immune response is still unknown. And on the other hand, on the host side, we find extracellular vesicles that are produced by both airway cells and by our immune system, and how those shape the bacteria, how they promote adaptation. We is still under investigation. We know that they contain or express on the surface neutrophil elastase. So that could be, a mechanism of crosstalk between the immune system and the bacteria itself by promoting adaptation. However, they also contain genetic material that can change how the immune system responds. And that's a talk that we will have tomorrow, on how this feed-forward mechanism changes neutrophil adaptation in the lung towards the phenotype that we see that is underperforming for bacterial clearance. So, all in all, there's still a lot of work to be done. I know I haven't touched upon the role of viruses themselves into how they shape the host pathogen interaction, and the work from Jan Bomberger, I think, is going to be critical to understand this relationship. However, the face of CF is changing, and how hormones are going to shape the immune system and how they promote chronic infection is still unknown in CF. Now that we're going to be faced with pregnancies, we know that the immune system actually is going to be more tolerant. And how does that going to impact the ecosystem in the lung and the microbiota? There was a really interesting study that showed that there is some tolerance induced in utero, actually, from the microbiome of the mother into the fetus. So all that is going to be open questions for our future. In addition to that, we're going to have an aging population, and we know that our immune system, as we age, changes and is not performing as it should, and how that is going to be in cystic fibrosis, we don't know. There is also a concept of trained immunity, and I don't know if Manu is going to talk about that, but we have a constant exposure to pathogens and viruses, and whether that is priming our immune system systemically and how does that affect other organs, we don't know. I think there is a lot more work to be done about that. Along the same lines, how CF-related diabetes and changes in, nutrients is going to shape how the bacteria reacts and how the immune system also deals with them. And in addition to that, we're seeing, emerging pathogens like non-tuberculous mycobacteria, and we still don't fully understand how macrophages and neutrophils, especially in the lung, are able to interact. And then the last 2 points are a little bit out there. We don't have much information in CF. We don't really understand how phages and human endogenous retroviruses actually shape our immune system. There are parallels that we can draw from other studies and other diseases that are not in CF. However, there is more research that needs to be done, as there are phages that can promote inflammation and phages that can dampen inflammation. We know in our preliminary that we can detect certain human endogenous retroviruses, and whether those are wiring our immune system towards an antiviral response. We don't know how that's happening, but I think that these are all open questions for our future. And the long story short is that there is a lot more work that needs to be done. So, yeah, I hope you all are prepared. And with that, I'm happy to take any questions.
As you are moving to the podium, I want to congratulate you on a beautiful talk, very comprehensive and concise and very easy topic for you to cover in the time frame allotted. And I also want to apologize to everyone, and to you particularly. I'd like to give you an introduction to our first in-person speaker. Okay. So bear with me. Sorry, Katie. Dr. Margaroli received her master's degree in medical biology at the University of Lausanne in Switzerland. She then completed her PhD in immunology and molecular pathogenesis at Emory University, where she studied mechanisms leading to neutrophil plasticity and chronic diseases with a primary focus in CF. She then completed her postdoc under the mentorship of Dr. Gagar at the University of Alabama, where she is now an assistant professor of medicine in the Department of Pathology. She is building a robust research portfolio. She's been recognized as a Blue Journal Emerging Investigator. Her work extends beyond the lung, also into the pancreas and into the pathogenic immunity in CF-related diabetes. So I think that's geared you up. I'm sure well-formulated questions now.
That was a really lovely summary of what we know about host pathogen interactions. So congratulations.
I want to apologize if your name was not up there. I thought of you. I just had to shrink it down.
This room is full of amazing people, so that's quite cool. So with that in mind, and all of the different players and angles and, subtleties there, you've thought about this a lot. What do you think about the concept of needing to subphenotype our patients so that therapies, particularly immunomodulatory therapies, maybe aren't right for everyone, and that we have to think about different aspects of this pathways you're showing might be more prominent in one group or another?
Absolutely. I was thinking about it this morning. I saw the program and I was thinking about the first time that I started here at NACFC, and I'm so happy that there are so many immunology related talks that are finally coming up. I think there is a lot more work that needs to be done in phenotyping the patient. The work that Dan is doing on, Th2s, type 2s. I think that we need to start to apply some of the immunology methods that we know and bring them into the field. I know Jay has been doing that for a long time, Manu as well, and Rabin, but I think there is a lot that can be learned and applied from other topics and bring them into the CF world. But I totally agree with you. We don't know exactly the granularity of the disease yet. And I think especially now with modulators, it's going to be even more variable. So absolutely.
Yeah, this is Jay Coles too. I was intrigued by the extracellular vesicle aspect of the work. And so in the GI tract, if you knock out IgA, you get extracellular vesicles from the gut microbiome into the portal circulation. and it causes liver inflammation. So, because presumably the IgA is somehow keeping the extracellular vesicles in the gut lumen and out of the liver. So do we know in the lung, are they coded by IgA or IgG, and does that affect the trafficking and then also who they interact with?
Yes. So I don't think that we know of that. What I can speculate is that the lung, so we see much more neutrophilic infiltration in the lung, and elastids can cleave both IgA and IgGs, making them inefficient for opsonization. So I would assume that we may not see the same mechanisms. However, I don't think that has been explored yet.
So I have one more. So when we think about how we're now treating these patients with ETI and it's transforming, obviously functional outcomes, much improved, but we still see ongoing inflammation, at least not returned to the levels that we would expect in healthy individuals. Now, you've invoked a couple different biomarkers that were slightly different than our classically thought TNF-alpha and IL-1 beta and IL-8. How are we going to sort this out in the context of complex inflammation that's going on? What advice do you have for us as we think about how to really target the immune cells?
I think that relates a little bit more to Catherine question as well. Like phenotyping. I think that we need to rely a little bit more onto the unbiased sampling. I know that is not pushed by the CF Foundation, but, with the reduction of sputum production as well, and as well the variability that we see across the lung. Because one study that I thought it was really cool that I didn't have the time to, cite is that different lobes have different host pathogen interactions that are going on at the viral level, at the bacterial level. So how do we get that granularity as well within the same one patient? So I'm a huge believer that we should start some BALs again, to be able to test and sample different regions of the lung. I know that, complications and, is not as pushed anymore. But I believe that that's a big step that we should take to be able to sample the lung, and get some consistency across sites as well. So that's the first step. And then I think that we should start to rely on, like lipidomics assay and proteomics assay and unbiased and embrace some machine learning as well in this, and see, how can we phenotype better our patients and have a full picture? But it's going to be a work for the long run.
We're restarting it. Okay.
Yeah.
Thank you.
No problem.
So our next speaker is going to be Emanuela Rashia, who is an associate professor in pediatrics at Yale, where she studies the role of CFTR in the immune system. Her particular focus is in macrophages, but she's really an expert in all ways. The current focus of her lab is to explore emerging therapeutic strategies able to target several aspects of CF lung disease, but primarily focusing on the macrophage. Her expertise makes her well suited for, I think, this section, which is going to be really discussing the potential immune targets for therapy in CF and focus on what aspects of inflammation are important and which ones of those that need to go.
Yes. Good afternoon, everyone, and thank you so much for the invitation. And Dito Camilla, I received this invitation and was okay. This is a very complex title and very complex topic to be able to cover in 25 minutes talk. So in preparing the slides, I decided to focus a little bit more on the areas that we are working in my lab that is related to the innate immunity and macrophage, but I will try to touch base on other aspect of it. So very exciting to be part of Game of the Thrones here. So we heard already the first two presentations and discussing how the immune dysfunction evolves with the disease progression. And when we think about the therapeutics, immune therapeutic strategies, we need to think in these terms. There will be not one therapeutics that will fix all, but this will really depend from the stage of the disease, the type of infections that the patients will have, and also the type of immunity, because let's remember about modifier genes that are contributing to the different immune response in patients with CF. So when we think about the immune response in CF, we think about this hyperimmune activation. In a normal lung, in response to infections, we have an inductions, we have resolutions, and the tissues go back to homeostasis. And this is lost in CF. We have this hyperinductions, the immune response that never resolve, and in the same times there is a fail to eradicate infections and overall a damage of the tissue. And so this brings me to the immune dilemma in CF, a disease that is characterized by hyperinflammations and effective host defense. Unfortunately, this is also true for people with CF that are eligible to highly effective CFTR modulator therapy, at least adult patients. This is, those are like some of the publications that are out there really highlighting how the CFTR modulator, although they improve mucociliary clearance, lung function, and they do not eradicate, they still have bacterial infections going on, and with that inflammation. And this brings to me why this symposium is so important, and in the game of the throne, we should consider the immune system a little bit more closely. So, we know a little bit the dysfunctions that are immune dysfunctions that are associated with CF. Of course, the mucosal barrier, the major innate immune defense that we have is extremely compromised in absence of CFTR. I would argue that probably this is the aspect that is a little bit more covered or addressed by CFTR modulator therapy. But when we think about the immune cells, we have dysfunctions that we have, over the year discovered in the context of innate immunity, in which players like neutrophil and macrophage, those are cells that should be able to sense the microorganism and destroy them immediately and help to repair the tissues. And we know that they are not working properly, failing to do their main function. When it comes to the adaptive immunity, we also know that there is an altered T cell response, and there is an imbalance in type of T cells that we see in the lung of patients with CF, with a predominancy in Th17 and Th2 response, and cells that are involved in keeping at bay the immune response, like regulatory T cells, they are down. And when we talk about humoral immunity, we would think about that in a context of a disease as CFO, that the bacterial is not killed, that the B cells will not produce the antibodies able to do that, immunoglobulin to do that. But it's the contrary. B cells are very active. They produce tons of IgG and IgA at the level of the mucosas, but they just do not kill bacteria. And Camilla discussed some of the potential mechanisms that are related to why those IgG are not, immunoglobulins are not recognizing killing the bacteria, but there are so many other questions that are open. Okay, Game of Thrones, the king, the winner of these battles are the neutrophils, I would say. There is no doubt that in patients with CF, this is the most abundant cells that we can see in their lungs, and they are central driver of lung damage and bacterial adaptation in CF. Camilla touched base on this aspect a little bit. They produce a high level of proteases that are destroying the lungs and are cleaving deteriorating aspects, molecules that are important to preserve the immune response. Also, they release mediators that, as Camilla discussed already, they may lead to bacterial adaptation. So there is not a surprise that those are the cells that for many years has been at the center of potential therapies for CF. And so one of the first clinical trials that tried to really address the neutrophilic inflammation CF, I'm pretty sure that people in this room is very familiar with this clinical trial. Those investigators trying to block the recruitment and activation of the neutrophil using LTB4 receptor antagonist. And this was not a successful trial. So actually, patients did had exacerbations of the lung symptoms, very much bringing up one major point that patients with CF are at high risk of immune suppression. And therefore, when we are thinking about a therapy for this disease, we really need to think very careful at how we are modulating it. Still at the center of the attention, again, I will not speak about the immunometabolism since Camilla discussed this a little bit. I want to just show with you one of the slide regarding blocking the granulation and proteases activity. We know that there are two major clinical trials for people with CF. Both of them are using small molecules that are inhibitors of cathepsin C. This is a protease that is produced by the neutrophils and is needed for the activation of neutrophil serine proteases, such neutrophil elastase. So essentially, those drugs are blocking the production of neutrophil elastase. And the results using those drugs are quite amazing in non-CF bronchiectasis. And indeed, the drug has been just recently, 1 month ago, approved from the FDA. And in the context of CF, there are ongoing clinical trials that are doing very well. So far, they are very promising. So hopefully you will have soon an additional drug in to treat your patients. Okay, now we'll switch gears and we will talk about my favorite cells, the macrophage, and why I think those cells need to be could be a good target for CF, for immune modulation in CF. They are driver of hyperinflammation in many inflammatory diseases, including CF. They are driver of pathogenic tissue repair. This has been shown in other chronic inflammatory disease, and I will show you some of the data that comes from our work that this is true also in the context of CF. And also we need to learn from other disease. Camilla discussed this a little bit. Let's think about the tumor-associated macrophage. In the tumor, we know very well that those macrophages are creating an immunosuppressing environment that limit the T cell activation and effector functions. And we do not know nothing about that in the context of CF. So is this something that we need really to evaluate or look a little bit carefully? And why I think those cells are fun to really target is that they are extremely responsive to local cues. So there are it will be easy to find molecules that will be able to switch them from a pro-inflammatory to a pro-resolving phenotype. And there are several studies that are exploring these directions, like the use of specialized pro-resolver mediators, the use of immune checkpoint inhibitors, or the modifying the immunometabolism. And also this, thinking about the future, those cells can be used for cell transplantation, for cell transplant. Indeed, this is something that is started to be a reality for diseases such pulmonary alveolar proteinosis. And kudos to Christine Haddock at University of Cincinnati that is trying to bring this technology in the context of CF. So, but which macrophage we want to target? I really like these old slides in which, in these studies, they summarized what happens during the development. So we are born with tissue-resident macrophage in the lung that they derive from fetal monocytes. And those cells are extremely resilient. They are able to self-renewal. They are able to deal with minor infections. They are able to keep tissue homeostasis. But as part of the aging, we are exposed to different stressors. Those tissue-resident macrophages are replaced by monocyte-derived macrophages, and they tend to be more pro-inflammatory and not very good phagocytes. So one of the questions is premature switch in macrophage composition in a CF lung. I know this slide is very busy, but, and I'm not going to discuss all those studies, but actually the literature that is out there is quite fantastic and goes back to more than 25 years ago. If we look at this literature, it is very clear that defect in lung resident macrophage function and composition starts very early in life. Prenatally, we just heard the study, the first speaker talking about how he can track this in the context of the pig model. And over time, with the developments of technology as single-cell RNA-seq, now we know that the migration of these monocytes recruited cells into the lungs starts very early in life. There is a very nice bioarchive paper doing single-cell RNA-seq in a group of patients, of children, preschool children. And then what happens when the disease is is established? And this is the work that Clemente Brito, the next speaker, will discuss. And at that point, there is the lung will be mostly populated by those monocyte-recruited macrophages. So what conclusion of this part of some of this part is that there is an early onset defect in lung macrophage function, and that the monocyte-derived macrophage increase over time in CF lung. I want you to pay attention to this immunohistochemistry. This was done on explanted CF lung, so we are talking about late stage of the disease. But here we were staining for CCR2 positive monocytes. So the CCR2 is a receptor that is expressed on those monocytes that are activated during inflammation. And is the receptor that the monocytes use to get to the tissue. And so, as you can see, all these brown stains, those are monocytes, and you can see how they accumulate in the parenchymas of those lungs and all around the scarring tissues and the lymphoid structure here. And so the point that I wanted to make here is that we know very little on what happens in the parenchyma of patients with CF over time, because we are limited to samples from airway secretions, but there is a lot that is ongoing in the tissue itself, and hopefully technologies such as spatial RNA analysis, sequencing will be able to address some of those points. Okay, I will spend the next couple of slides talking about some models that we have developed to study this specific problem and the switching between the tissue resident to the monocyte-derived macrophages. And this is work done by Ravindra Gunapanavar. He's a postdoc in my lab, a talent and postdoc in my lab, that will discuss some of this work tomorrow. So he developed these models in which we sprinkle wild-type CF animals with a little bit of LPS for several days, and then we let them to recover. And we do that in wild-type CF knockout animals. As you can see here, doing this procedure in the animals, we can see the number of tissue-resident macrophage increasing over time, and this increase is blended in the context of the CF knockout knockout animal. But most importantly, when Ravi looked at the BRDU, so we are looking at proliferation, what you can appreciate here that in wild type, this white bar, the wild type animals, those cells are proliferating, and in the CF they are not. Okay. And also, those cells are much more prone to apoptosis. But the question is, what is the ontogeny of those cells? And to answer this question, we have developed another mouse model in which we can track the cells that are differentiating from the CCR2 monocytes. And when they are coming from those cells and they will differentiate, they will maintain a fluorescent protein that in our case was the M-cherry. So let's see this in actions. If we look at those cells in wild type and CF animal at 5 days recovery, what you can see here that those tissue resident macrophage, while in a wild type are mostly not deriving from the M-cherry population, in CF we have 16% of the cells that are deriving from the monocytes. So definitely now we have a tool in which we can study why this conversion is happening and what are the tools. Understanding that the mouse model do not fully characterize recapitulate the the pathology in CF of CF. So the conclusion here is that in a normal lung, we are exposed to stressor, but we have very resilient cells that are populating our lung. And so with those stressor, although we lose some of them by cell death, they are able to proliferate and repopulate the niche with little bit that comes from the monocytes. But in CF, we have early stressor, and this is the mucus flakes, the hypoxia, early infections that are cleared, the lack of CFTR, and all together, those initial stressor may lead to depletion, may lead to less replication, and therefore repopulation of those monocytes in the niche. But does really matter? Are those monocytes really bad or they are working properly? And this is one and another example when a mouse model can be informative. In this study, what we did to address this question, we developed a model to study how neutrophilic inflammation, chronic neutrophilic inflammation is damaging the lung. And so we took wild type and CF mice and we treated them with low doses for 5 weeks. And so we can see that at the end of 5 weeks, there is much more damage in the CF lung compared with the wild type. And to answer the question whether the monocytes may contribute to this damage, we took advantage of the mouse genetics, and so we crossed the CF knockout animals with the CCR2 knockout animals, double knockout. And if you remember from the previous slides, if you lose the CCR2, the monocytes cannot reach the lungs. So just few piece of data from this study that we published a couple of years ago. The double knockout, if we look at the lung damage score during at the T1, so at the end of the chronic exposure to LPS, the double knockout are protected from lung damage compared with the CF. And this is another piece of data that really led us to scratch our head and was totally unexpected. They are not just not doing better when it comes to the lung damage, but also in general, they are doing much better. Here, I'm showing the weight loss of those animals in response to LPS. CF animals, they lose more weight during any type of stress that you get to them. But if they are in the double knockout, in the CCR2 knockout background, they essentially are doing much better. So a lot to learn of how inflammations may impact really the whole well-being of the animal. So if I need to think about a therapy to use macrophage for rebalance immunity, really will depend on the stage of the disease, right? If it is the beginning of the disease, we need to target, make the tissue resident macrophage to be more resilient, right? Increase their fitness and able to deal with the initial infections. However, later on with the development of disease, probably those cells are not anymore there. And so we need to find a strategy to make the recruited cells better phagocytes to fight the war, right? And I do not do justice here. There are so many people out there that are working on those aspects, and there are so many strategies that people are testing in preclinical studies. And so I apologize to not go through the all potential line of investigations, but a very exciting time. Okay, so I'm fine. Okay. Another unjust here. If I knew that I had the extra minutes, I would dedicated more time to this. But potential strategy for targeting adaptive immunity in CF. And when I think about this, I put this icon because that is an aspect in CF that is really understudied, really, and there is so much to learn. Right, so we are up here. We have discussed this imbalance of T cells, and Camilla discussed also the data that comes from Christine here and the J. Cole group trying to target the Th17 2022 Th22 response. But I wanted to spend like 1 minute discussing also some of the work that has been done by Dan here, our co-chair, and looking at exuberant Th2 immune response in CF. So from several studies that he published, he have shown that people with CF that have an exuberant Th2 response, they have a worse prognostic. And therefore, there is also the need to target this pathway. He also has some basic science, basic biology studies, very much exploring how the CFTR in T cells is impacting the signaling, the interleukin-4 signaling, so changing the activation of T cells. And this is very exciting. And this is very exciting because if it is true that this is a pathway that at least for a group of people with CF could be therapeutics, well, we already have biologics for that, right, in the context of other allergic diseases. So there is a very interesting perspective of having a potential therapy for those patients. And finally, I wanted to spend some time about B-cell activation. Again, another underexplored area of investigation. We know that very little about what happens. B cells are activated. There are, again, tons of those IgG that are produced and immunoglobulins that are produced. Camilla discussed how the neutrophil elastase may degrade them, and so there is not opsonizations and phagocytosis, but also we are learning a little bit of the basic mechanism. I want to point out a recent work by J. Coles in which he started to look into that in details and realizing that using mouse model that the IgG are there, but they are not all the same. They are all skewed to a specific outer membrane lipoproteins that is present in the Pseudomonas aeruginosa. There is a difference in the type of IgG that are produced, and so is this something that can be targeted? Now that we started to identify and study the mechanism, we can think about how to immunomodulate, and there are the field is evolving very fast in potential technique that we can have, approaches that we can have to target those problems. I didn't have time to add. I removed those slides, but because the time. But I wanted also for you to think about the immune defect and whether this is rooted in differentiation. This goes back to the first speaker that we had in sections looking at myelopoiesis and how this is impaired in animals, piglets, so at birth. Some similar work that we are doing in collaboration with Diane Krause at Yale University and Cassia Braga, another postdoc in the lab. She will discuss this in poster 3665 if you want more information. But really, we are interested in looking how the chronic inflammations, right, is cross-talking with the hematopoietic stem cells. Those are the cells that are producing the all blood cells, including the immune cells, over time, right? And so we know that chronic inflammation is inducing changes in these hematopoietic stem cells. And as Cassia is presenting in their poster, when we look at epigenetic changes by ATAC-seq in hematopoietic stem cells of CF mice compared to wild type, after chronic exposure to lipopolysaccharide, we see major epigenetic changes that are related to the production of an effective immune cells. So we need to think about this circle, that when we arrive to the late stage of the disease, it's not just what happens in the lung, but some of the dysfunction are imprinted at the level of the hematopoietic stem cells. And so, again, probably targeting only the cells in the lung will not, at the end of the story, help a lot at this stage. So I'm concluding. The general conclusion is that immune target for therapies must be tailored to the disease stage, the type of infections that the patients will have, and immune response. This is exactly what Kate was discussing, personalized immune therapies. And to do that, we need to solve point one. We need to advance our understanding of CF-related immune dysfunction. And then I want to also make this other point to conclude here. We are in the perspective of fixing the disease with gene therapies, and this is great. But to do that, we need to go back to point one, because one of the major barriers that we will have in a gene therapy field will be exactly overcoming the CF immune barrier. And so with this, I thank you for your attention. I presented some work from people in the lab, and so I really deeply appreciate all of you, and also my collaborator at Yale's, because they make my life so happy. So with this, I'm happy to take questions.
So I have one question to get us started here. Thinking about the neutrophil studies that you were showing, and you referenced that the CF population might represent an immunocompromised population in which we need to think heavily about the therapies that we're giving. Is this changing now with modulator therapy, and might it be possible to revisit some of these more immunosuppressive or targeting therapies now that we have the protection of ETI within a subset?
Yeah, absolutely. This is going back what Camilla just discussed. These omics studies will be extremely important at this point to trying to understand what to target and how the disease is changing. And we have some good amount of studies that are related adult patients, so patients that started the modulators when the disease is already established. So let's see what will happen with, studies that are coming out from younger children.
That was wonderful, Manu.
Thank you.
So you talked about why macrophages are good targets, and I completely agree. But you mentioned two things, and then you presented some really compelling data, and I want you to synthesize it. So you said that they are plastic, and you can shift their phenotypes, and you mentioned that they're long-lived. You're just talking about ETI and changing things, but you showed really beautiful data that Cassia Braga's doing, that you're getting epigenetic changes. So are those going to remain post ETI? And really, is our macrophage targeted therapy going to be macrophage transplantation or bone marrow? If you get these epigenetic changes happening, can we actually shift those cells to the right phenotype? Or once those epigenetic changes happen, is that too late?
Yeah, well, yes. Reversing epigenetic changes. Well, for what we have seen, no, you will not it will be very difficult to reverse them once they are established. For some of the work that we do with the mouse models, we can take the hematopoietic stem cells with epigenetic changes and transfer to a normal host, and we still see those cells that are not working properly. So I would say that will be very much complicated to be able to reverse those memories, right? What I think that it will be important again is to trying to I think the sweet spot is starting like before those epigenetic changes are really accumulating into the host. Yeah.
Yeah, thank you for a fantastic talk. I was also wondering your thoughts on how we can better model, especially in the animals, some of the other chronic factors that may be impacting our long-lived cells, such as our factors from our diet, the Western diet, environmental factors that, macrophages are uptaking and reshaping their profile. Because, we can do it in short-term studies and human samples, but how do we think about that better? Because there's many of these things that are reshaping that.
Yeah, that is a very difficult, very difficult to address because, of course, animal model can teach us certain point, and then, and if we think about, I am a little bit distressed about the idea to take everything in vitro, like the NIH is pushing us to do human studies, which are informative, but when you want to kind of dig into mechanisms, you really need to have models to do that you can manipulate with a control. And also the in vitro system is limited. I mean, we see it's not just two cells crosstalk. We are speaking about crosstalk between the GI, the bones and the lungs. So we need to rely to animal models, I think, at a certain point to understand the mechanism. And to get close to the disease, I think that we can learn from the mouse models, large animal models that we have, like the ferret, the pig, maybe they can, with specific question, they can a little bit help to move forward and understand how this can apply during the process of CF. Yeah. I hope I answered your question, kind of.
Thank you for a really nice talk. So my question is kind of related to alveolar versus interstitial macrophages. So you showed alveolar macrophage data, and of course they're typically, obviously they're tissue resident and they self-renew on a steady state, but interstitial macrophages is typically monocyte derived. So I was wondering if you've compared, the amount of infiltrating monocytes that are going towards interstitial versus alveolars, and then beyond that, if you've managed to subset them at all, and then what your thoughts are potentially targeting alveolar macrophages specifically or interstitial macrophages separately for therapies.
Yeah. So in some of the work that we have done, we have looked at the interstitial macrophages, very interesting population. And yes, they are deriving from the monocytes, and during infection and chronic inflammations, what do? They accumulate into the lung of the CF animals, and these accumulations match is a lot compared to wild type animals. So there is those accumulations of interstitial macrophage, and if you think about this, those are the cells that will perform a lot of activation of T cells, right? And again, this is going back to my point that I made in one of the slides, that we always focus on the airway secretions, so the cells that we are able to see in the airways, but in the interstitial space, there is a lot that is happening that's contributing to the disease. And again, those cells are the cells, those interstitial macrophages are the cells that are going to do a lot of crosstalk with the T cells and that activations. And so we are studying a little bit, going in that directions, because we see differences between the wild type and the CF animals will be difficult special transcriptomics probably can help, but to look a little bit into the human situation of human lungs and from people with CF. Yeah.
Thank you.
Thank you.
While Dan is pulling this up, I have the pleasure of introducing our last speaker for this awesome session, Dr. Clemente Brito. He's an associate professor of medicine and physician scientist in the Division of Pulmonary Critical Care and Sleep Medicine at Yale. He has many roles, including attending in the medical ICU. He serves as the associate director of the Yale Adult CF program and the director of the Yale Pulmonary and Critical Care Medicine. I'm sure it will be there. Fellowship program. Surely these activities complement his research program focused on understanding the role of the airway epithelium in the development of airway diseases. The goal of his research is to define the role of sex on immune transcriptional activity that drives sexual dimorphic clinical outcomes and responses to therapy. His translational work, as I'm looking up and
You just keep.
I'm just going to keep on. I can keep on going, fortunately. But his translational work on air.
My homework.
Well, his translational work, while we are plugging this in, on airway biomarkers and transcriptomics in CF, including studies in single-cell profiling of the CF airway immune responses using both mass like not the technology favoritism at CFC.
Oh, hey, we're good. Oh, we're good. Once that's still caught yet. Well, it's just my luck to follow Emanuela and Camila on a tour de force review of immunity, so I'll try not to disappoint. Thank you so much, Christine and Dan. Sorry for all the mess with the presentation. So, my role here now is to try to bring a little bit more into perspective of how we look at all these data, all this discovery that we've made over the years, and give you one example of how we can begin to apply it to personalized care and to personalize our understanding of innate immunity into some more directed, phenotypically directed approaches to therapy. So I'm really excited to share some of the work I've been working on for the past year, on sex-specific innate immunity networks of the CF airway. I have no conflicts of interest in these skills related to this talk. And I want to start by showing you a slide about patient differences. Maybe not. There you go. All right. So, yeah, no, so we're going to make it right. I promise. So our patients behave very different. We basically take two patients with CF, one with the same mutations, right? F508 del homozygous. You take two of them, you treat them with the same stuff. They need the same course of treatment. They have the same social environment. They still behave differently. And it's really hard to understand where that comes from. There are many reasons why that happens. And in our lab, we're interested in the sexually dimorphic part of that difference. So historically, people, before the modulators came along, there was a very clear sexually dimorphic presentation for males and females with CF. So females with CF tended to have worse lung function, more exacerbations, and worse clinical outcomes overall. They also tended to acquire more pseudomonas and males. So I'm going to show you here an example of the trajectory of two of our patients. One female, one male, same F508 del homozygous, same age, same therapies, and how differently they behave in response to therapy. So a female patient here starts at an FEV170, which is a little bit lower than the male that was CF. Then she hits Trikafta, a huge bump in lung function, about a 50% increase in lung function. There, on the other hand, is a male comparison here. He starts with a slightly higher lung function, which is consistent with an untreated male before modulators, has a little bit better lung function. And then he hits Trikafta and he doesn't respond, right? But he has a flat response to the cephalema. This is not necessarily representative of the entire CF population. This is just an example of how variable the courses can be from two patients taking the same drug with the same underlying. In terms of their microbiology in sputum, for example, this female patient had Pseudomonas and Staph in sputum. Took a long time after clinicians changed their flora. Our male patient only had Staph, so consistent with their clinical phenotype of less Pseudomonas acquisition, monthly. And then he transitioned from Staph slowly towards Hemophilus parenthesis. So two very different clinical courses. And it just gets us thinking about how, even though we understand CF is a monogenic disease, we still understand that there's a lot of variability and that his presentation is actually quite heterogeneous. While CFTR function remains the main determinant of the clinical features of the disease, there's a lot of other genes, mainly immunity agents, that are critical modifiers of this clinical force, this clinical response to therapy. And also, the reason why we're all here is because there is an extensive immune dysfunction that has been quite well characterized in CF. And that extensive immune dysfunction comes into play into that force and that therapeutic response. So, what we've been trying to do is very simple and a very loaded question, which is why are the CF manifestations of our patients? And I have to give the caveat that this genomics and biology focus explanation, which means that it really doesn't take into account the complexity of a CF patient. If you ever care for a CF patient, you know there's a lot more than biology involved. But so we have our CFTR, that is the main driver of these clinical features. We all can live with that. But then there are always other disease modifiers, right? And those can be host defense, innate immunity, etc. The expression of those heterogeneous across cell populations and sexes and locations. So in the end, the net effect of the effect of the residual CFTR function aggregated on your modifier genes, aggregated on your cell populations, the expression of your cell populations determines a heterogeneous clinical course of heterogeneous therapy. So the dream is that you can go to a point where you can identify these patients according to their cell populations, so the gene expression, and then obtain a more homogeneous response to therapy, or perhaps a more homogeneous clinical course overall. So I've been on this quest to develop a framework or a process that is non-invasive to characterize and immunophenotype our patients so that then we can separate them into groups that we can understand and treat differently. And I've done that with sputum. Sputum is awesome. So sputum is awesome. Sputum is a non-invasive sample that occurs at the site of the pathogenic process itself. So blood is accessible. The blood has not received, the blood is not having the interaction. Cells from the blood are not having the same interaction as the cells in the lungs. Cells in the lungs are interacting with bacteria, low pH, dead cells, dams, PAMs, all those things are located right there, and they're directly transforming the transcriptional profile of those cells. Important, different from the cells in the blood. The cells in the lung have undergone transmigration, which confers an extraordinary transformational shift in their transcriptional shift in their overall gene expression profile. So they're completely different cells before and after they're going through there. So we use different incentives to motivate people to participate. Sometimes we're a little short on staffing, so we may hire whatever is available in the hiring pool. And but the job gets done. And then in our lab, depending on what we're doing, we focus on the cellular or the cellular component of the sputum. So the first step that we took here was to try to understand the transcriptional differences in the sputum samples of people with CF and people without CF. And it was a very basic study, essentially. These are the people that do all the work. That's just blah, blah, blah. Provide some live entertainment. But really, these people were the people that did all the transcriptional analysis, sputum processing, all the hard work here, all the heavy lifting. And these other gems here that came after are people without whom this project could not have been completed. So we generated a process to cryopreserve sputum samples, separating those cells, and then that was a significant improvement because we were able to pool our samples to decrease variability. So then after they're all frozen, we have a big batch, and we thaw them, filter them, resuspend them, and eventually end up with a single cell suspension that runs through single cell RNA sequencing. I imagine that most of you know more single cell RNA than me by now. So I'm going to skip the description of what it is, but let's just say that it is a process of identifying all the gene expression profile within individual cells that are attached cell by cell to their patient, to their origin, and to all the clinical information that we can query from that connection. So this is what we found initially. So we found something really cool. We proved everybody wrong that sputum is actually a good sample and that you can actually detect every immune cell that is present in the sputum. You can profile them with quality data. So we identified eight different immune cells. So perhaps more clearly outlined here, if you ask in your analysis, show me the cells that came from a stress patient, show me the cells that came from a healthy control in green here. You can see that there's quite a distinct pattern between the two. So in people who are healthy controls, the vast majority of their cells are in green. There are alveolar macrophages, right? These guys are here. Whereas our people, the CF, have either neutrophils or this really interesting population previously not well characterized of monocytes. So there is a ton of monocytes in the airways of our CF patients. And that is something that, even though the neutrophils are like, yeah, they have purulent sputum, the monocytes were quite different. So we decided to focus on that for a little bit and just try to characterize them a little better and understand what the meaning of that new population was, which Manu already kind of gave you a spoiler alert that they are actually important. So we took those cells and we used a different kind of single cell analysis that is called pseudotime and then archetypal analysis. Essentially what that is if you think about getting a sputum sample, you grab a sputum sample and all the cells came from the lungs to the, but they are at different stages of maturation. So we plot them along the trajectory of how many mature cells or less mature cells you have along in that sample. It gives you a sense of what is the typical trajectory for those cells to be in. So then we identified two CF specific trajectories of maturation within those monocytes and macrophages. Importantly, those two subsets of monocytes were either pro-inflammatory monocytes that were very immature, very angry immune cells that could not cure bacteria but caused a lot of trouble, or heat shock activated macrophages, which were a little bit more mature in terms of looking like a macrophage, but were not effective at clearing bacteria either, and exhibited a lot of issues with cell death pathways. Meanwhile, our healthy controls had a preponderance of just healthy, pro-resolving, bacteria-clearing, mature monocyte-derived macrophages. So, as a summary here, these are things to remember. So, we identified several immune cells there. We had two unique CF-specific monocyte macrophage pathogenic archetypes. But the thing that we really wanted to do there, we could do, is that it was a small study. So I couldn't really study what I want to study, which is the differences between subsets of patients that we can profile. For example, males versus females, or treated versus untreated, so that we can identify the difference that makes a clinical presentation different. So we hunkered down, collected a ton of people, and collected a ton of money to be able to do some single-cell RNA sequencing. But it was worth it, I would say. And we're now beginning to answer some of those questions. So the main initial question that we're trying to answer in terms of disease variability, or what makes two groups of people different with CS, is sex. So looking for sex-specific differences has been one of the ways in which we're trying to explain this clinical variability. And what we did here is we used single-cell RNA sequencing to profile the cells from males and females. And what you're looking at are gene expression heat maps. So these are essentially graphic representations of how many of the gene expression profiles in our patients. So each gene, each row is a gene, each column is the average of expression of a patient. And then you have separated here each individual cell type that we identified in the lungs and males and females, right? And without paying too much attention to what the genes are right now, you can tell that there is a remarkable difference in the gene expression profiles of each individual immune cell within that airway, whether it was macrophages, alveolar macrophages, neutrophils. And then if you look deep into what those genes are, you'll find some of your favorite genes in there. NACFC, IL-1 beta, CSF3 receptor, interferon signaling mechanisms, etc. So, getting to that point, then we can ask even more sophisticated clinical questions. Like, for example, I want to understand what makes high exacerbators different. And so high exacerbators, like people that exacerbate more than 2 times a year, there's people that don't. And then is there a sex difference? Is there a treatment difference? So here I'm showing you an example of taking from those high exacerbators only the monocytes and looking at their expression profile between males and females. So what I'm showing you here essentially is that if you take the people that are in the hospital frequently with CS, their gene expression profile is not the same for males and females. So if you wanted to treat a high exacerbator with a high monocyte count, you would target IC15, for example. So those would not be effective if you were trying to modulate that immune system in females with a high monocyte count, where instead you would be targeting or row A. So we're now talking about focusing therapy on individuals that may be female patients with a high monocyte count or male patients with a high monocyte count. And their approach to treatment would be different. I have really cool data on pseudomonas, and lung function CTs. Reach out if you're interested in hearing more. So, getting at the other part of really interesting clinical subsets that I'm interested in is response modulators. So do these modulators really change the gene expression profile of our immune cells in the lung? And the answer is yes, it does. They do. And this is data a little bit older than the rest of the data that I'm presenting, but here you see people who are either treated with Symdeko or Trikafta, and then on the other hand, people that are not treated. And what you can see there is that each immune lung cell that you see here has a different gene expression profile according to their treatment with modulators. So modulators in themselves elicit a transformation from what the no treatment profile looks like to the actual treatment profile, which we have fondly come to call transcriptional reprogramming. That's not what I'm interested in. What I'm interested in is if you take people and treat them all with Trikafta, do males and females remain the same transcription? I think it's much more exciting data because we show that males and females may improve with Trikafta, but the reason why they're improving is not the same. Well, males and females may not improve with Trikafta, but the reason why they're not improving is not the same. So when you're trying to address that discrepancy or that lack of response to the treatment, you have to try to focus in a little bit more on how can we address targets, the sex-specific difference might make them different and might make them important. And now I'm going to talk about something that is really interesting. And, if Camila was talking about strategies and different houses in the Game of Thrones, and I'm going to talk about the gossip in the palace. And so what you're looking at here is something we call analysis. So think about it this way. What we're looking at is we're trying to understand how I will communicate with you, right? So there's different cell types over there, but we're trying to understand my outgoing signal. So I'm going to send X amount of ligands. So that's outgoing signaling. That's my ligand gene expression. And then you over there are expressing receptors, some receptors variable for those ligands. So that's, we're going to have income signal. When I produce a ligand that interacts with your receptor expression, there's a potential interaction between the two to create a connection. And that connection over a number of expressions will then generate a connector, an overall overview of how a group of cells will communicate with each other. Yeah? Okay. So, a couple of things. So you're comparing the group of healthy controls to the group of CF patients. The first thing you see in the words of the great Marie, is a circle on the right looks a lot more fussy than the circle on the left, which translated to connectome lingo means that there is more ligand receptor interactions per cell type. So you see that? Right. So the monocytes send more signals. But they also, but the cells in CF that will interact with those monocytes express more receptors, so there's more potential for it. But there are also signaling mechanisms, signaling networks that are built between those monocytes and these sets of T cells. So, for example, for your work, guys, in adaptive immunity, you're seeing here that those networks that are there, the blue and the brown ones, for example, do not occur in our healthy controls. So they're CF-specific disease networks. And also, networks that are anti-inflammatory, like non-alveolar macrophages, for example, use a healthy control. And you can see that red network over there is absent from our CF patients. So you can see there, and if you wonder, for example, what is in that red network, IL-10 signaling, hemoxygenase 1, so anti-inflammatory mechanisms. So we have different mechanisms that are transforming and not present in some of our CF analysis. And this is the most exciting part of my data, and it's only a couple slides. Perhaps the most exciting now in the era of highly effective modulator therapy is what does this treatment with CFTR modulators that we now know alters the programming, the transcriptional programming of our immune cells do to our actual networks, to our actual overall interactions. And this is something really exciting to me. So what you're seeing here on the left is connectome plots of pro-inflammatory cells from the lungs in untreated and treated people, everybody, males and females. So without going through all of them, just because we really like monocytes in this room right now, you can see here that our monocytes from CF are expressing a ton of ligands and a ton of receptors. And then they get treated with Trikafta over there, and there is a drastic reduction in the overall network formation and the overall predicted interactions between those monocytes and all those cells. So that is really important, right? Because the fewer networks you build, then the fewer things you have to target. So, and then it happens with monocytes and macrophages, with neutrophils and macrophages, monocytes are the coolest cell. So then, now this is really cool, because the other were inflammatory cells. The inflammatory cells are experiencing a downregulation of the overall signaling before and after Trikafta. These are alveolar macrophages, pro-resolving, bacteria-clearing, homeostasis kind of cells. And what you see here is that they're not going to sleep, they're getting active. So I didn't put the picture here, but if I show you the treatment plots of an untreated versus a treated person with Trikafta, their repertoire shifts back from neutrophils and monocytes back to alveolar macrophages. So not only do they increase their alveolar macrophages, those alveolar macrophages are active. They're energized. Oops, sorry, going here. Okay. Last slide. So even more exciting is that if you look at it from a sex-specific difference perspective as well, you will see now that there is the same phenomenon that we saw before is now happening again, just in a perhaps more dramatic way. You can see an even more dramatic reduction in the outgoing signaling from those monocytes after Trikafta, and you can see it in the others as well. I didn't say it in the beginning, but this is I only picked the interleukins, because I can't put everything I want to put into a single circle, right? But you can iterate this with integrins, with interleukins, with interferons. Like it's infinite amounts of data that you can process depending on what your area of interest is. And then here in females as well, you see that same reduction in the signaling, or perhaps, that activation of that network building between it. So just as a summary here, we went through these modifier genes and how we were looking at those modifier genes within the context of cell-specific expression. But perhaps instead of looking at cell-specific expression, what we should be looking at is network-specific expression. So rather than like a back alley stabbing for the Lannisters, we're talking more about a red wedding kind of situation when you treat, right? Like we're talking more about targeting an entire network that is critical for the signaling rather than looking for a gene. And then again, looking at it from neutrophil-rich females, monocyte-rich males, etc., trying to target not only the subpopulation of patients that you're looking at, but also looking at it from the perspective of a group of networks that are critical for that presentation. And then hopefully we'll get back to that dream of getting a more homogeneous trajectory here. So these are things that I think are worth remembering. So we have immune expression that is sex-specific in CF and is associated with these dimorphic clinical features in some cases. CFTR modulators do induce transcriptional reprogramming phenomenon here. And that reprogramming that we see is sex and cell type specific. Finally, it looks like these immune signaling networks in the lung are also sex specific and modified by Trikafta, creating really great opportunities for personalized medicine and for immunophenotyping of our patients. So I'll leave you with that. I want to thank thank you guys for inviting me. Thank you for hearing me talk a little bit about our work, and to all our patients and our contributors for all their help and their support, and especially our collaborators everywhere. Thank you guys. Hey, Kristen, how you doing?
Superb talk. That was great. I just wonder if the mapping tells you anything about the crosstalk between the immune cells and the epithelium, if you have any sort of key pathways that are ligand receptor interactions.
Yeah. So yeah, I have it. It's just that when we do sputum analysis, if we get a lot of epithelial cells, we kind of see them as a contaminant, right? Because it means that you shouldn't really have all that many epithelials in your sputum. So if you were doing a brushing, for example, then that would be different. But these are cells that for one reason or another fell into the airspace where they shouldn't be. So we do have the data and we can build some of those connections. We just don't know how to properly interpret that within the context.
Thanks. That was a lovely talk.
Thanks so much.
How much of the male-female difference is due to estrogen? And if it is due to estrogen, do you think that will change in the female CF population as they get older?
Yeah, great question. So when you think about these sex-specific differences, the first obvious thing that comes up is it's the hormones. Right. So that makes sense. And there is very clear documentation that estrogens, for example, are inducers of pro-inflammatory behavior on monocytes, macrophages, and neutrophils, whereas progestins and androgens, for example, tend to be somewhat immunosuppressive. Also with the caveat that estrogens are supraphysiologic conditions, for example, in the cases of pregnancy, for example, can have an immunosuppressive effect as well. So there's sex hormone mediated regulation of some of these genes. Now, there is non-sex hormone mediated gene regulations. For example, there are genes that induce interferon gamma at different levels that are sex chromosome related. We have chromosome silencing, like TLR7, for example, that we silence one copy of our TLR7 so that we don't get an autoimmune response to it. So there are many non-sex hormone mediated mechanisms through which you can also see some of these effects. So we're working on that now. So what we're doing now is we're trying to, beyond the in vitro human cell model, we're trying to go into a more in vivo model that actually reflects immune response and can have a better example of that regulation in a mouse, for example. I guess one more tidbit of information might be cool for you to hear is two things. If you take, say, a macrophage from the brain, the lung, and the peritoneum, they're going to have very distinct transcriptional profiles. And those transcriptional profiles are sex-specific as well. The other thing that's important is that they express sex hormone receptors at different rates. So I don't have a slide here, but if we look at the sex hormone expression for estrogen, androgen, and progestin in macrophages and monocytes, for example, they're different. And that may also condition a little bit of the sex hormone effects. Sorry, that was a very long answer, but I hope it was good.
Well, I kind of had a very similar question, but do you have any sense of like people with CF that are taking ETI and they go in hormone replacement therapy? Like now that people are, living longer, like how that would change, or if it's something that you're planning to study?
Yeah, so it's really challenging because you have to actually pair your sex hormone measurements with your sampling, right? So we're working together with Raksha Jain and Jennifer Taylor-Kauser. We're putting together a project where we would actually look at those sex hormone differences in menopause, in sex hormone replacement. But it's a challenging undertaking because you really have to you have to do it all at the same time, right? Like you have to collect the sputum, collect the blood, and then interpret that in context. But we do have data on, most of our patients, whether or not they were menopausal, not menopausal, whether they were taking hormone replacement or not. Like we do have some of that, and we're making efforts to collect more data as we go forward. Thank you.
Okay. So first of all, I would like to thank and congratulate all the speakers for this fantastic session. My question for Professor Brito is this one. Have you, having this wonderful data set, have you tried to compare with the available database, like for example, last year I believe a group from Marcus Mall published the effect of ETI on immune cells taken from the nasal swabs.
Yes.
So if you see any similarities, or because that can be another option to collect immune cells from the airway rather than the sputum, which, as you said, it's difficult.
That is actually a great question too. So the short answer for those that may not be familiar with this, we can actually integrate data sets even if they come from different sources. There's a lot of computational massaging that has to happen for that to actually happen, but we can get a relatively representative merging of the two samples when we do that. So this particular data set was done in younger patients from nasal brushings, and then in the nasal brushings they analyzed separately the epithelial part and then the immune part of this. So we're interested in the immune part, and at some point we're going to get to that. For us, it's a really valuable proposition, more from the perspective of validating. So it's two things, right? One is validating the data set. So that what I find really replicate what you find. And then also how deep down the airway can you go before your data are not really mergeable or, uniform anymore. Like that concept of a unified airway where, if you sample here, it doesn't matter if you sample here, right? And my expectation, at least in earlier mergings that we've done with other data sets, is that not upper airway, but if we compare BAL sampling versus sputum sampling, at least, we do see a very comparable cell population distribution, except for alveolar macrophages, because you are washing down there. You can't beat that. Yeah, so you'll get more of those. But but that is a really important part of the work. So we have that in the docket. We just haven't got to it yet.
So maybe I have something that you also already have in the docket, which is like what Camilla suggested, to use a machine learning approach like random forest to identify the top decision-making point for there can be also therapeutic targets for the immune shaping the immune response in CF.
Right. No, we love machine learning, right? And here is, this makes a lot of sense because you have so many potential connectomes to identify, right? And what we look for in principle is, the thickness of those lines, of those connecting lines, sometimes can give you an inkling into how strong the relationship can be. So the edge weight for us gives us a clue, but really you have to analyze this on thousands and thousands of piles of data. So it does make sense to train a machine learning model to then identify the critical
simplify the model.
Because we love TNF alpha and IL-1 beta, but maybe that's not where the money is, right? So it is important to also have an unbiased approach to try to identify and discover those pathways. Thank you so much. Those are really cool questions.
Thank you all. All right. Please join. Well, let's have that after the talk. You can come up and ask him directly. Okay. Thank you all for coming, and please join me once more in thanking our speakers.